So I'd like to thank the organizers for being wonderful again to invite me to come here. And it's been a great privilege to come to Bangalore each year, actually. And so I'll start with my talk, which is on random metrics and network approach to complex system. I changed the title a little bit from before. And here are my collaborators, Pradeep, Rakhi, and Pooja. And I will be talking about work that we have done together. So here's the plan for today, and in the interest of time, I'll skip this and go on to the motivation, in which I'll be discussing two systems, the biological networks, proteins, and here, and the climate system, in which I'll be discussing the El Nino problem. So in the biological networks, I'll be talking about proteins, and here are some proteins which we found in the web. We will not be able to talk much about the alpha and beta helices in the protein, but we will be able to tell you the important positions of the proteins, that is the hotspots. Now, in the context of the climate system, which is we look at the El Nino problem, which I'll discuss with you, and there we will try to predict the El Nino climate phenomena. And there also we will be able to detect the geographical positions which are important. Okay, so we turn to biological systems, and I'm flashing this slide to remind you that proteins will be taken as a string of repeated amino acids, which are 20 in number, and the amino acids determine proteins. The sequence, the order actually, the sequence of amino acids are important for their three dimensions, shape, and the structure of the protein determines the function. And here, the reason for using data is that the experimental methods are very difficult and expensive, and so data is a good method to complement experiments. Today I'll be talking about this particular protein family, the IGPD protein family. I was talking about the beta-lactamase for a while, and these protein families are present in plants. Okay. And what they do is actually cut the carbon-oxygen bond in certain metabolic processes involving making histidines. Histidines are another protein, okay? And they're used in the immune system. And here is a snapshot of the crystal structure and the reaction, the chemistry behind the action of this enzyme or protein, IGPD protein. So what we will be interested in doing is finding in these proteins the hotspots, okay? Using a very technical and statistical mechanic method. Okay. So that's what's written over here. And this is how we do the data and data filtering. So we go to the raw data is found in the PFAM database, and it has this entry number. And this is the IGPD protein. It consists of almost 6,500 sequences. And you do filtering analysis and get it down to 487 sequences. And also there's another filtering that needs to be done because there'll be gaps. Okay, so that has to be also taken care of. And here is the multiple sequence alignment that is done in these data sets in which similar characters of the amino acids are put into line. So that's how you do the data. And then you find out the correlation metrics. Okay, and here is a distribution of the correlation elements, okay, for different properties. Okay, so instead of the amino acids, you put properties, okay, like the hydrophobicity, the relative mutability, polarizability, and the polarity. Okay, and so this is the distribution of the elements. Okay, so you can diagonalize that matrix and get an eigenvalue distribution given here. And you can see that there are certain eigenvalues that are outside the bulk. Okay, and what you can do is to shuffle the column and the rows, and you'll find that there is a bound. It becomes a random Wishard matrix, and there's a bound which Archishman talked about, the Markington-Pasture rule. So this correlation matrix, if you shuffle the rows and columns, becomes a random Wishard matrix with these bounds. Okay, and the bound for this particular matrix, correlation or the Wishard matrix, is 2.43, which is about here, and the smaller bound is at 0.18, which is given in this inset, which is around here. So you notice that there's several eigenvalues outside the maximum bound and the minimum bound, and they are 35 in number. Okay? And those carry actually the system-specific information about that system. Okay? So that's what we generally focus on. Okay? And so you get different eigenvalue distributions if you put in different properties. Okay? There were 4 properties. If you put different properties, you get different eigenvalue distributions. And you can also calculate other spectral quantities like the inverse participation ratio and the entropy of eigenvectors. And these are the formulas corresponding to that. And these are the figures. Okay, so this is the inverse participation ratio. This is the eigenvalue. And you notice that the random, the Wishard matrices gives you this red line, whereas the hydrophobicity, the IgG protein for the hydrophobicity property gives you this blue. Okay, and you see that most of the contribution is from the blue, from the lower or the smallest eigenvalues, not the largest. Okay, and similarly, if you look at the Shannon entropy, you find that the smallest eigenvalues give you the large contributions, whereas the largest give you less. And it turns out, if you analyze it a bit, that the smallest eigenvalues are very highly localized, and they're highly localized in comparison to the largest eigenvalues. And that's given here in this diagram, in which one is plotting the square eigenvalue components on the y-axis, and in the x-axis, the positions. And you can see that the blue corresponds to the smallest. So these are very localized, whereas the largest are localized, but not as much. Okay, so from here you can say that the positions, okay, at which the localization happens, okay, gives you a sector. Okay, this is the sector, and these are the hotspots. Okay, so for the first eigenvalue, we found these hotspots. For the fourth and second, we found those. Okay, and okay, so we can go ahead and do a network analysis. I will go quickly through the network and show you this network. Okay, in which we're taking the hydrophobicity and the network when the threshold changes. That's what I wanted to know. The threshold changes, it changes, the network changes, and for a certain threshold, you get the same hotspots. Okay, and that's where you stop. Okay, so yeah, and you can do the same for the other properties, and you get different structures. Okay, so, and here is a diagram of the particular PDP, particular IGPD protein in which the hotspots are given over here. And if you target those, you can control the action of this protein. Okay, I just wanted to touch on the climate system. That's why I needed a few moments. We looked at the El Nino phenomena, the climate phenomena, and I need a few more minutes, but this is an advertisement for maybe next time. And here is a timeline of the El Nino, N missing, El Nino episodes. And this is So this affects, okay, this El Nino climate system affects the weather from all the way from Australia to South America and beyond, okay? And so it's very important. And these are the, we've taken the data of these climate, okay, the temperature data of this system, okay? And one finds if you do a network, one finds in the normal year that the network is very connected, and in indicator year, which is 190 in 1981, it becomes less connected, and in the El Nino year, it becomes again densely connected. And this is a indicator of El Nino and can be used to predict it. Okay, if you take some other quantities like the largest node and so on, the links and cluster coefficient, they also decrease in the indicator year and increase again in the El Nino year. And the eigenvalue statistics also has certain, if you take the number of eigenvalues outside these bounds, the Mackinger-Postures bounds, you can see that the number of eigenvalues increases during the El Nino. And you can also do the inverse participation ratios. You find that the smallest eigenvectors are highly localized, again. And here, from there, you can find out the important positions. You can see in the indicator here, the important positions for the smallest is this, and the networks, it is here. And for the El Nino, it increases, okay? But it is at the equator. So at the equator, the El Nino, should be studied. And in other El Nino episodes, if you look at and analyze those, you find that the positions that are important are the equator and the tropics. With that, I thank you very much for your patience.
Thank you, ma'am. Let's have one quick question from the audience.
Hello? So this random Mushat eigenvalue bound, so you're using that as a way to screen.
It's just like Archima had said, as a null model.
As a null model, just to make sure, so no correlations here, anything above or beyond
whatever
that.
is there in the, within these bounds, or which correspond to that, act like a random.
Right.
So, and the eigenvectors are extended. Okay, outside it, they're localized.
Right.
And those give you the sectors and those the structure of that system. And you can use this method. I mean, you can't use it always. It has to be sufficiently complex. And the sufficiently complex is to see whether there is a null model. Okay, the Wishart matrix is there. And yeah.
Let's thank the speaker once again.
Thank you.
Okay.